All right, we are introducing you to the concept of biology. Biology is the study of life. That's what makes it different than chemistry. Chemistry is the study of matter. So biology is the study of living things. So it's matter also, but living things. So bio is life. So when we talk about this life, if you haven't taken a biology class, you can walk through a field and say, oh, look at all the plants, or walk through the forest and say, look at all the trees. But as you become more and more educated, you start to see things that your brain wasn't aware of before because you didn't have the knowledge. And there's a famous phrase that says, the mind doesn't know what the eyes can't see. So as you learn more in biology, it's going to open up the world to you a little bit more. You're going to see the diversity that exists out in the world, and you'll look at your landscape a little differently, I hope, as a result of this class. This class has a big environmental focus, so you get a little more awareness of how your way of living impacts the environment, and you can help spread the word to others to do simple things that have positive impacts on our environment. So diversity is a big thing you're going to talk about all semester in this class, and in spite of that diversity that we see here, you know, there's bacteria, a paramecium there's a fungus, plant, and an animal in this slide. You know, they're all living things, and they're very diverse in their form and function, but they share similar characteristics. So when you look at the characteristics of life, there's several characteristics that you should recognize that all living things have, regardless of how simple they are. And one is that they are organized. So our organisms have specific structures in them that allow it to perform specific functions. So they're very organized. It's not just random pieces of matter and cells smushed together. You know, they have specific functions, and they're very organized. So when we look at the organization of life, starting from the most simple to the most complex, all living things are made up of atoms. So atoms are the most simple form of matter, but when it comes to living things, atoms come together to form molecules, and the molecules come together to form cells. So that's where we step into biology, because atoms and molecules are simple forms of matter that we study in chemistry, right? And those are not living things. So when we come into the biology level, the simplest form of living things are cells. So all living organisms are made up of cells, and some might be a single cell, like the paramecium in the previous slide. That's one cell. So that's the basic unit of all living things are cells. So like the slide says, unicellular or multicellular. Unicellular means it's a one-celled thing. Bacteria, made up of one cell, one bacterial cell. Paramecium, amoeba. And some of those things can be really nasty, right? Amoebic dysentery is a very severe form of GI illness, causes a lot of diarrhea and death in third world countries. Number one cause of death in children worldwide is diarrhea. Very sad, because it's easily treatable here in the United States, right? But in third world countries where they don't have access to clean water or medicines, children die from diarrhea. So when we look at the structure of organisms, it tells us a little bit about their function, and we'll go into the details about that when we get into the specific systems and organisms. So the atom and the molecule, like I say, are the simplest forms of matter. They make up the cell. So the cell is where we start to get into living organisms. And then different cells come together to form a tissue. So the building blocks of tissues are cells. For example, fat, adipose, is a tissue, and it's made up of fat cells that all look pretty much the same, right? They're all, if you look at fat on chicken or burger, it's the white, right? It's the white stuff that we see. And if we looked at it under a microscope, we would see there's individual cells in that fat. Muscle tissue is made up of protein fibers, and those are all specific muscle cells. And then we go and look at different tissues. Those make up organs. So if we look at, for example, the heart is made up of different tissues. There's muscle tissue that makes the heart contract. There's connective tissue on the outside that holds the heart in place. And there's a real thin inner lining of the heart that keeps it smooth and slippery to allow blood to flow through, with very little friction. So different tissues making up that heart. And if we look at a leaf, there's different tissues that make up the leaf. So the leaf is an organ of the plant made up of different tissues, and we'll talk about that in lab. And then those organs make up an organ system. So we're getting up here in our level of organization. This is way more complex than the lower levels. So the heart and the blood vessels make up an organ system. The stomach, the pancreas, the liver, small and large intestine, rectum, mouth, esophagus make up the digestive system. So that's an organ system made up of many organs. And then all the organ systems working together, like in the human body, the respiratory, nervous, and all the other systems working together make up the organism. So on a lecture quiz, we might ask, what do we call a group of cells working together? What would the answer be? Tissue, yeah. Which is more complex, an organ system or an organism? More complex would be an organism, because we start from the simplest with atoms and we work our way all the way to the most complex, and that's the organism. Okay? So those would be some questions that you might see on a quiz, just asking you to describe each level of this level of organization or be able to rank them, which is most complex. And that's a really nice diagram you have, and that came from your textbook, so you can look at that there too. And then if we continue on into this organism, it goes even further. Organisms, let me see if I can make this a little bigger here. There we go. So we're at the level of the organism. If I have a group of organisms working together in a specific area in the environment, that makes a population. So a population would be like a group of fish in the ocean. They're all the same species. They're living together in the same area, a school of fish, right? That's a population. It's all the same species. It's the same organism. But then if I take a bunch of organisms in a specific part of the environment, for like in this picture here, we have elephants and zebras and the trees. That makes up a community. So those are different organisms. So a population is the same organism. A community are different organisms, but they're living in the same environment. Call that a community. And then if I look at the water, the temperature, some of those non-living factors, we call them abiotic factors, along with the living organisms, that makes up an ecosystem. So we're really getting complex now. We're going to look at an ecosystem. And then different areas of the world are broken up based on weather and other factors. Landscape, mountains versus oceans, grasslands versus ice patches or ice, ice sheets. Those make up the biosphere. So lots and lots of diversity when we get to the level of biosphere, right? If you look at atoms, that's very low diversity, but it gets more and more complex, more and more diverse as we go through this list. So again, be able to define each of these terms for our lecture quiz. A lot of information there. And we're going to be working our way through this. We're starting our first unit here, once we're done with this introductory information, is looking at chemistry. So we're going to be starting here, and then we're going to talk about the cell. And then we're going to go into the different things that cells do. So a lot of the class is looking at complex processes like DNA and protein synthesis, cellular respiration, photosynthesis. It's not until we get to the last third of the class that we start to look at the different organisms and talk about ecology. So when we look at populations through biosphere, that level of organization, that's a lot of ecology, and that's at the end of the course. So we start out pretty heavy in this class in terms of complex things. So don't get discouraged, just keep working hard and know that as the class gets toward the end, it lightens up a little bit, and then we're more into those things that you're more familiar with and you hear about on a regular basis. Okay, so characteristics of life. So all living things share some things in common. And one thing is, whether it's a bacterial cell or an elephant, they all need to acquire nutrients and energy from the environment in some way. How do plants acquire energy? Where do they get their energy from? From the sun, through photosynthesis. And animals get their plants from, or get their energy from other organisms like plants, right? Some break down chemicals in the environment. We have bacteria that live like in the sulfur beds in Yellowstone, for example. There's bacteria in there that use chemicals to acquire energy. And we'll talk about all those different types. But just know that all living things need to acquire nutrients and energy from somewhere outside of their body. So autotrophs are plants. They get it from the sun. They can make energy outside of ingesting other organisms. And then there's heterotrophs like us that need to get our energy from other energy organisms, energy consuming organisms. So autotrophs are plants, and heterotrophs are animals and fungi that need to get their energy from other energy consuming organisms. Homeostasis is another characteristic of life. We need to maintain an even keel, and we have systems, feedback systems in place to make sure we're at an even keel. For example, temperature. We operate best around 98.6. The human being does, because our enzymes in our brain depend on a 98.6 body temperature. And if we go too high, feedback systems kick in, and what happens? What does the body do to bring temperature down when it's really hot? We sweat. And our capillaries of our skin dilate, so our face gets red, right? So we bring that heat from the blood to the surface by dilating capillaries at the surface. And that's when people drink too much alcohol in the wintertime. Alcohol causes dilation of blood vessels at the skin surface and causes rapid heat loss. That's why when people have been drinking and they pass out in the snow, they're more at risk of hypothermia because of that abnormal feedback mechanism of the alcohol causing dilation. They lose heat really fast. And as a nurse, I've taken care of people that have passed out in the snow after drinking too much, and sometimes they lose their fingers and toes because of that, because they get frostbite, that the skin dies and they can't repair it. When we cut ourselves, we have feedback systems where the tissue releases platelets to block the little cut that we just made, and that stops the bleeding, like a paper cut. Stops bleeding pretty quickly because of the feedback system of those platelets being added to that little tear and clogging that little damage to that vessel and stopping bleeding. So homeostasis, hugely important. When you eat a meal, your body responds to that high blood sugar in your blood by secreting insulin to bring your blood sugar down, because we can't have chronically high blood sugar. We need to get that sugar to our cells to feed our cells and keep us going. If you have low blood sugar, your pancreas releases glucagon to raise your blood sugar. Like if you didn't eat breakfast this morning, you're not passed out from low blood sugar because your pancreas is secreting glucagon to raise your blood sugar. And some, more common with young women, have low glucagon release, and they end up having low blood sugar, and they get kind of shaky and sweaty if they haven't eaten in a while, and that's just because of that feedback system not working quite right. Yes? Yeah.
So is that what the
Exactly. Yep. Low glucagon secretion. Yeah. Very common in women. And I have that issue, so I have to always like eat a little snack, or if I eat something really sugary for breakfast, that spikes blood sugar, brings it really high, and then you release a ton of insulin, which crashes your blood sugar really low, and then you get the shakes and the sweats. So people that have hypoglycemia, it's recommended that they have a protein source for breakfast, or a multi-grain type of breakfast. Not a donut, unfortunately, not white bread toast or a bagel. Those things can spike blood sugar and then drop blood sugar. So it's good to have some protein like scrambled egg, or just even a whole grain cereal that has a little more polysaccharides to it. Okay, another characteristic of life is responding to stimuli. Here we can see the sun is coming in this window and the plant is bending toward that sun to maximize its energy in the leaves. So it's responding to the environment. If we didn't respond to our environment, you'd come to class today with bumps and bruises and bleeding open sores because we are constantly turning over in bed, you're shifting in your chair, you're leaning forward, you're leaning backward, because you're responding to signals from your body that it's getting tired of being in that position, right? So that's responsiveness, and we need to respond to our environment to keep ourselves warm, free of danger and pain and injury. Reproduction and development. We wouldn't be here if our parents didn't reproduce, and even within our cells, we're constantly producing new cells. Like your red blood cells die after about 3 months. You need to make new ones. Your skeleton replaces itself about every 10 years. Your skin replaces itself every 30 days. Right now, there's skin cells all over your bedroom and your bed and your apartment. If you have lots of people that you're living with, the dust you see on your furniture are skin cells that are being shed from everybody in the environment. So it's a good reason to dust, right? So you're not inhaling all that, but our lungs are good at keeping ourselves healthy too, well, in most cases. But that's the importance of vacuuming. Vacuuming and dusting keeps the skin cells under control, and the dust mites don't eat as much as a result of that, because dust mites in our environment feed off of skin cells. So we're constantly replacing things. Reproduction helps us pass information on to the next generation. So survival of the fittest, you've heard that before. So organisms that are healthy and strong are more likely to reproduce than organisms that are weak and unhealthy. That doesn't really apply to the human being, though. We have excellent medicine, so people that are normally infertile can still reproduce because of help they get with fertility, or people that have chronic diseases, with treatment, they can be strong enough and be able to reproduce. But animals fighting in the environment, like deer, for example, the weak ones that are thin, aren't going to survive the winter. And in areas where there's a high deer population, where there's a lot of competition for resources, we see, crowding, overcrowding, and disease. What's the big disease here in the Midwest with the deer? CWD, yeah. There's a lot of issues with that, especially in Minnesota. Everybody has to get their deer tested. Some people like to, kill the deer and then eat the heart right away, right? Well, they don't recommend that anymore, because if that has chronic wasting disease, it's not a good idea to be ingesting that until we learn more about that disease. So everybody should get their deer tested. Some people poach and take a deer and don't tell anybody. They're putting themselves and their families at risk by eating that deer that hasn't been tested. So we'll talk about DNA this semester, so we'll talk more about the details on that, but just know that being able to reproduce and develop is a characteristic of all organisms. And then adaptation, being able to adapt to the environment. So over time, as environmental conditions change, we're going to talk about at the end of the semester how a group of finches on an island were able to adapt to the changes in the weather patterns on that island, and how the vegetation changed, their beak shape and size changed, so they could utilize different food sources and survive. So adaptation takes a long time for that to happen. Evolution is, hundreds of thousands, millions of years of adaptation and change over time. So we are going to study different organisms, and we can group organisms into 3 major domains. So maybe you learned in high school, like when I learned it, it was King Phil cried, oh, for goodness sakes. Kingdom, phylum, class, order, family, genus, species. Do you remember learning one of those in your biology class? Okay, so the first one starts with domain. So there's 3 major domains of life. The archaea are special bacteria that get information from chemicals in the environment. So they're a little special. They live under extreme temperatures, extremely high and extremely cold temperatures. So they're in their own class, they're in their own domain. And then the bacteria are in their own domain. They are unicellular organisms. Very simple, they lack a nucleus. So archaea and bacteria, they both lack a nucleus. So make sure you write that down, that they lack a nucleus. Prokaryote means no nucleus, no organelles. those first two columns there, or rows, I should say. And E. coli are a good example of bacteria because E. coli is everywhere. It's in our digestive tract, it's in our stool, and that's why when people get food poisoning, E. coli is the #1 culprit of food poisoning related to people not washing their hands after they use the bathroom and they go back to work. It's the #1 cause of urinary tract infections in females, and it's their own E. coli on their body that is infecting the urinary tract. So it's a big problem in human health. And then the eukarya is anything that has a nucleus and organelles. So we can see that paramecium there, even though it's one-celled, it's unicellular, it has organelles and it has a nucleus. So it's more organized than the bacteria. So those we fall into the domain of eukarya. So they have a nucleus. The first two do not. So we're going to spend a lot of time in these first two looking at those, and we'll look at bacteria. We'll grow bacteria in lab. We'll throw some antibiotic discs on some agar plates and see if we can kill bacteria with different antibiotics. And that'll happen about mid-semester. So just basic introductory information. So the next topic then is moving into the scientific method. And we're going to do that with our lab reports. You're going to really analyze the scientific method, go through the scientific method with a lab that we're going to do together in class. And the #1 step in the scientific method that you should write down, it's not specifically on the page, but the #1 step whenever we design a scientific experiment is we identify some problem in the environment. And when I say it's a problem, a math problem is not a problem, right? It's not like a bad thing. It's called a math problem. It's something that needs to be analyzed and solved. So when I say identify a problem, we're just looking for something, some observation that we want to ask a question of. So the first thing we do then when we identify the problem is we ask a question. For example, does coming to class affect grades in biology? Does attendance impact grades in biology? That's a question. So that's identifying the problem. The problem is grades and attendance. Is there a relationship? Okay? Again, when I say problem, it's not a negative thing. It just means something we're going to explore. So that's step #1. And then the next step, #2, is to make a prediction. And one would hope that attendance does affect grades, right? So coming to class, students who come to class will have a higher course grade than students that don't come to class. Or I could be more specific, and because we want to make your hypothesis measurable. So if it's something that is measurable, it means it's quantitative. It's some dimension, it's some value that I can record. So what would be a good way to write a statement about the relationship between grades and attendance? My prediction is students, what would you write? What would be measurable and easy for everybody to understand? Because when we do a scientific experiment, it has to be understandable by anybody after the fact and repeatable. They can replicate the experiment and get the same result if it's a true experiment with valid data. Do you have your hand up? Yeah. Okay. Who miss? We could even be more generic with those numbers. You chose 1 in 4. How could we narrow that down to be more easy to gather data? Because you could have like 2 columns of data, right? Yeah, very good. Who miss more than 4 days of class will have what? We could say a grade percentage. Then those who miss less than 4 days of class. So this is a good hypothesis because it's measurable. What am I measuring? What am I going to measure? Yes. Am I going to measure that? What? Well, okay. When we look at this, there's variables. What are the variables? Let's identify that. What are the variables? We have an independent variable and a dependent variable. So the dependent variable is what I measure. The independent variable is what I'm going to adjust in the experiment. I'm gonna separate my group based on this variable. What? Yeah. That's what I'm separating my group based on, is attendance. And I'm gonna measure the grade percentage, yep. So when we look at any experiment, there's always a dependent variable and an independent variable. And now you could flip these around and say that the other one is the dependent variable and the attendance is the dependent variable. We have to say what determines what. To figure out what's independent and dependent, we have to say, do grades determine attendance? If I'm an A student, does that make me come to class more? You could look at it that way, but physically, if we talk about hours of sunlight and plant growth, we could keep it more simple than that, more simple like that. Does the sun affect plant growth, or does the plant growth affect the sun? Right, so the sun is the independent variable. It doesn't depend on anything. The sun shines, right? In the experiment, we're going to set the attendance, and we're going to see if the grades depend on the attendance, right? So and this is a really important concept. Students get the dependent variable goofed up all the time. Just think, the dependent variable depends on the independent variable to happen. So the grades depend on the attendance, correct? That's what our prediction was, right? Is the grades depended on the attendance. Okay, so so next to the independent variable, I want you to say, to describe it, is that it is changed to test the dependent variable. So we change the independent variable to test the dependent variable. Does that make sense? So I'm going to change the amount of sunlight to see if plant growth is affected by that. Okay, in that very simple example, I'm going to change the amount of sunlight. So I'm going to put them under plants, or I must put some in the dark maybe, and I'll put some in the sunlight, right? Or whatever I'm going to test with light. I'm going to change the independent variable to test the dependent variable. And it's real easy to figure the dependent variable because the dependent variable is what's measured. The dependent variable is what's measured. Make sure you're writing this down because I'm going to give you a crossword that uses these exact terms that I'm giving you to describe this stuff. So the dependent variable is what is measured, and it depends on the independent variable. That's why it's called the dependent variable. It depends on the independent variable to change. So the plant growth depends on the sunlight. And we're going to measure plant growth. Okay, so the grades depend on the attendance, and we're going to measure the grades over the semester and compare that to the attendance, the greater than 4, what would you say, 4? Greater than 4 and the less than 4. That's what we're changing in the experiment, right? We're taking my groups and we're saying, here's the greater than the 4 and here's the less than 4, and let's measure their grades over the semester. Okay, then we need a control variable, a standard variable. So whenever we conduct an experiment, for example, if I want to say this medicine, this protein shake builds muscle in weightlifters, where if you take this protein shake and lift weights, you're going to have more muscle than someone who doesn't drink the protein shake and lift weights. So who's my control group then? Yeah, the people who don't drink it, right? And this is really important because when you look at a new medicine, for example, they say, oh, this is really helpful with depression. And they say, you know, we tested this medicine and the placebo group, 30% of the placebo group had less depression compared to 35% that took the pill. Is that enough to take that pill? If 30% felt better just because they thought the pill was working, and 35% felt better that were actually taking the pill, then you'd say, that doesn't convince me to take that pill, right? And that's why you've really got to be good consumers. So understanding the scientific method is really important for anybody, not just biology students to understand, but the average consumer to understand. You know, for example, when we look at the vaccine dilemma, those who vaccinate have less death and disease than those who don't vaccinate. Is there data to support that? Huge, huge amounts of data, huge amounts of data. But when you have Sally's healthy blog on the internet saying that my son got autism right after the MMR vaccine, oh my gosh, I'm not going to vaccinate. That's not looking at data. Anybody who looks at data can see the relationship between the independent variable and the dependent variable and the control group. So it's just like when you buy a car too. They can say this is the best car ever, and then you go online and look it up and they say worst car ever, you know, had all these problems. But if you take everybody who drove that car and see who enjoyed it and who didn't like it, the majority would support the car, right? Because of data that the automobile manufacturers are gathering, not just the blogs or whatever on, you know, if you like your car or not. So it's really, really important when we're making health decisions for ourselves to know the experimental design and what it tells us. So the control variable does not get the treatment, okay? And then control group is they're not exposed to the experimental variable. The test group is those that are exposed to the experimental variable. And then replication. Replication means, can I repeat this experiment and get the same result? So when you hear the word replicate, I want you to write the word repeat after it, because it has to be repeatable. And the guy that came up with the autism is caused by the MMR vaccine, he actually lost his medical license because he came out with this big experiment and made this big conclusion, and everybody was all upset and, you know, not giving their kids the vaccine. They went and tried to repeat his experiment, and they found that he had botched the data, and he lost his license, his medical license as a result of it, and they squashed that whole experiment and said that it was false. But so many people took off with it because they wanted an explanation for autism that it's still out there, that the MMR vaccine causes autism. And they're saying that it's this preservative in the MMR vaccine called thimerosal that causes the autism. And actually, thimerosal has never been in the MMR vaccine. So it's just such bad science, but people are making conclusions and changing their health decisions based on that. Yes? Measles, mumps, and rubella. Yeah, and so kids get that about 1 year. And what they don't recognize is when kids get about 1 year is when autism starts to show itself. So autism is kind of a late in development, you know, in early baby development to really show itself. Oftentimes doctors will see early signs and they'll say, well, let's wait till the 1 year visit to make a decision. You know, let's wait. And if babies aren't talking, they're not responding to the environment, they're seeing different behaviors, then they say, yes, you know, we're starting to see that development. So, and is there something in the environment that's triggering autism? Because we see a lot more autism nowadays. Definitely, definitely. But we can't just, you know, if I drink a can of Coke and get hit by a car on the way out of class, we can't say drinking a can of Coke causes auto death, right? So we have to have good data and repeatable experiments to make our conclusions, and we're not there yet. And when doctors themselves are vaccinating their own kids, they have access to the data. It's not like the doctors are these bad people that are keeping a big secret and just vaccinating everybody to keep the medical or the pharmaceutical companies happy. They're vaccinating their own children and themselves. Because I ask that. When they present new things to me, I always say, what are you doing? How are you dealing with this or that? And they'll say, well, I take this myself, or I've done it myself. Like one of my kids needed Accutane for severe acne. And I looked online, oh my gosh, it looks like you're drinking gasoline with what they said online about the side effects of Accutane. And I was like, oh my gosh, I'm not gonna let my child take that. And then I talked to some people and they said, they're giving this to kids that are on chemo, have bad livers and bad hearts. They're giving them Accutane and getting good results. Like, okay, well, when I look at the data supports that overall this is an okay drug. So my son took it and his face is clean as a bean and no side effects. But with anything, there are side effects and not everybody responds the same. Okay, so this is just an example of an experiment. We're exposing them to light. So we talked about the hypothesis, independent, dependent variable, control variables, experimental group, and how could we replicate that? So what would the independent variable be? What are we changing in the experiment? Very good. The amount of light would be the independent variable. And the independent variable is always placed on the x-axis. So make sure you write that down on your notes next to independent variable. This is the x-axis, this is the y-axis. We always put the independent variable on the x-axis. And this is going to be important for you to know for your lab reports. And the y-axis, that's where the dependent variable goes. What would the dependent variable be? Yeah, the growth rate. And we would have to be specific. Are we going to measure that in centimeters, inches, meters, whatever? Depends on the plant. And the control variable, what are things we'd want to control? We don't want to add extra things into our experiment, right? We want it just to be plants, plant growth, and light. Yes? Okay. The control variable, we want to make sure that it's specifically 7, specifically 8, right? Specifically 6. Okay, what about type of light? Can I put some in a window or some under UV light in an artificial lamp? Would we want to control the light source? That's a control variable, right? We don't want to have different light sources in there, right? And that's a real problem in educational data that I always raise my hand, and I'm the troublemaker in the group. When they say, if we do this, students do better. Well, but do students who do this, are they more likely to be a better student, so therefore they did that? And they're already a better student? It wasn't necessarily that caused it. So it's really, hard to control variables with human beings because there's so many factors. But yeah, so how about the pea plant? Should they be the same starting size, right? Same species of pea plant, right? What else would affect plant growth? Yes? Very good. Yeah, soil composition, amount of water given to the plants should be the same. Control variables, right? Yeah, temperature, another control variable. So we have to think about that too when we look at things that are impacting our own health decisions. What are the other variables? Have they been controlled in the experiment? Because there's a lot of bad science out there and it's impacting your decisions. You read it online, you think, oh, that sounds great. Really look at the details. Control group. What would be the control group?
The plant exposed to 6 hours of light.
Sure, yep. You could set that as your control variable, or you could even say less than 6 hours. You could have a group that is 5 hours. Experimental group would be the ones that are the increasing, right? We're got to finish up here. And then accounting for replication, you have to repeat it, right? So we need to repeat this and hopefully get the same result. We're not going to get the same exact result, but we should see the same relationship, right? More light, taller plants. Okay, the next step then. So what we're doing, so first we identified the problem, we made a prediction, and then number 3, we design the experiment. And when you design the experiment, that's when you're identifying your variables, identifying the control variables, experimental group, test group, or control group. And then we have to analyze. So when you design the experiment, this is when you're gathering data. Data is what you're measuring, right? Then we come up with analysis and conclusions. For analysis and conclusions, you need graphs and tables. To analyze, you need to take your data and put it into something visual to look at the graph or table and look for results. Then you formulate an idea. And the word formulate is going to be on your assignments. And make sure you write that, formulate. So after you've undergone analysis of your data, you come up with conclusions and you formulate an idea saying, yes, sunlight, increasing sunlight does increase the growth of plants. And then from that, you can decide if you're going to repeat that or dismiss it and say this was a bunch of junk and there's no relationship. And sometimes that happens. So a theory is something that based on our observations, we say we believe this is true, but we can't repeat it because it's not something that's repeatable. Like we can't repeat evolution. We're here at the end of it, or maybe in the middle of it based on, another million years. But all we can do is look at the environment around us and say infer. The word infer means to come up with a suggestion and an idea based on observation. So we can infer that we think this happened, but we can't create a scientific experiment. We can't repeat it, so it remains a theory. What are some other theories? The theory of relativity from Einstein. We can't create different time intervals. All we can know is the time interval that we're in, right? And traveling at the speed of light, we've been able to calculate the speed of light, right? And everything is relative to another thing, but we can't necessarily observe that because we can't zoom out and look at the whole universe. Maybe there's another universe out there, right? All we know is where we are. So we make observations based on that. When we look at other experiments that are repeatable, and we see the same thing every time. I drop a feather in a vacuum and it falls at 9.8 meters per second every time. That's the speed of gravity. Is everything falls. For every second it falls, it speeds up 9.8 meters per second. So if I drop a penny off of the Empire State Building, it's a really tall building. Every second it falls, it's speeding up by 9.8 meters per second if there's no wind resistance. So let's say there was no wind resistance. That penny would go into the concrete. If you held your hand out, that penny would go through your hand because it's traveling at such a high speed with gravity. And we can repeat that. Newton's 3 laws of motion. For every action, there's an equal and opposite reaction. We can repeat that in an experiment and get the same result. Law of conservation of energy. That is force equals mass times acceleration. We can repeat that in an experiment. So these are things that are repeatable, observable, no problem, they become a law. These things are a theory, so we're always gathering new information and they can change. So what I want you to bring back on Wednesday is this crossword on the scientific method. Yeah, today's Tuesday, yeah. The words are on the back to make life a lot easier. So if in doubt, if it doesn't fit, you know it's the wrong one, right? But I want you to be comfortable with these terms because some of them will be on your test.
Thursday, though?
Thursday, yeah.
Okay.
Bring it on Thursday, yeah. Normally I teach this class on Monday, Wednesday, so I got to change my thinking.